Welcome, everyone. My name is Zhou, and I'm a researcher in the lab of Dr. Emma Gutman. I am delighted to share with you all our publication on behalf of Dr. Kazu Yamamura and the other incredible researchers who are listed here. Now, atopic dermatitis, which I will call AD from now on, is driven by Th2 inflammation. However, the mechanisms that sustain this inflammation and impair the immune regulation, which should be keeping this inflammation in check, are not fully understood. The OX40 and OX40 ligand pathway is of particular interest, with OX40 expressed on activated T cells and OX40 ligands on keratinocytes and antigen-presenting cells, and this interaction promoting T cell survival. Also of note, there are several therapeutic antibodies targeting this pathway that are in clinical trials. So our study aimed to understand how the OX40 ligand axis is contributing to AD pathogenesis. Our findings have reaffirmed its effects on effector T cells and uncovered new effects on regulatory T cells. In our study, we examined the skin and blood samples from AD patients and healthy controls using the multiple modalities that are listed here. And our key results are as follows. So firstly, OX40 and OX40 ligand are highly expressed in AD skin compared to healthy controls. Through immunohistochemistry and immunofluorescence, we found that lesional AD skin has a much higher infiltration of CD4, CD8 T cells, and Tregs, as shown here, especially in this bar graph. Furthermore, the proportion of OX40 positive cells in these immune subtypes is significantly higher in lesional AD skin compared to healthy control skin. And this is especially true for our Tregs right here. Now, interestingly, AD patients also have elevated OX40 expression in circulating immune cells as well. Specifically, in AD patients, CD4 T cells, effector cells, and Tregs that are CLA positive, which is a skin homing marker, basically meaning they can enter dermal tissue. They also show a significantly elevated expression of OX40 compared to healthy controls. In this particular graphic, the AD patients are the black dots and the healthy controls are the unfilled dots. And the CLA positive cells are listed here as well as here. So interestingly, the proportion of OX40 positive Tregs is also actually positively correlated with AD severity. Now, so far this data would suggest that there's a pathogenic role for OX40 in atopic dermatitis. So next we aim to establish a possible mechanism for its effects. In the next set of experiments, we activated PBMCs from healthy controls and from AD patients, either in the presence or absence of OX40 ligand, to see how or if the expression of Th2-related genes was directly modulated. Interestingly, by RNA-seq, we found much higher expression of Th2-related genes in the AD patients compared to the healthy controls. Now, as expected. However, in the presence of OX40 ligand, the Th2 gene expression may be bumped slightly, which you may be able to see from here to here. The black bars here are AD patients once again, and the healthy controlled patients are the unfilled bars. So the Th2 expression may be bumped slightly, but not really significantly in any group. And these findings were confirmed for both qPCR and ELISA, in addition to the RNA-seq. So then, how might the OX40 pathway be exerting its effects if we don't see a signal in the effector CD4s? Well, remember, it was not only the effector CD4s who showed abnormally high expression of OX40 in AD. The regulatory T cells, the canonically immunosuppressive T cells, did as well. So when a similar experiment was performed with Tregs, this is again purified from PBMCs, then stimulated either in the presence or absence of OX40 ligand, we see a significant downregulation of IL-10, arguably the most important immunosuppressive cytokine expressed by Tregs. And this effect, notably, was only seen in the AD patients, not in healthy controls. So in conclusion, we found that OX40 signaling may weaken Treg functionality, therefore allowing for the persistence of Th2 inflammation in AD. And with that, on behalf of Dr. Yamamura, the Rockefeller Institute, and Dr. Gutman from Icahn School of Medicine at Mount Sinai, I would really like to genuinely thank the editors at Allergy for allowing us to share this data, and thank you all for listening to this talk.